we discovered a new feature of SARS-CoV-2, which was entirely unexpected. It links physiology, the disease, and the virus itself. It enables us to rethink strategies and to come up with a new approach to combat and defeat the virus. When the university went into lockdown, we changed tack completely and started to become virologists, trying to figure out what's going on with this new virus threatening us all. SARS-CoV-2 is the virus which causes the COVID-19 pandemic. It is the virus which infects us and causes this terrible disease which has cost countless lives. So we know a lot about the structure of the virus. We don't know a lot about the mechanism and why it is so dangerous and what makes it different from the other coronaviruses. Some of them are just circulating.
So spike proteins are on the surface of the virus. They are the proteins which are known to be crucial for infection of the human cells. They dock onto the receptors or proteins on the cell, and they help the virus to enter into the cell. And the mutations on the SARS-CoV-2 spike protein are the ones which are thought to be responsible for this higher infectivity of the SARS-CoV-2 virus. So initially for us, the aim was to generate spike protein and to help establishing serological tests. We decided to quality control our spike protein, and we used cryo-electron microscopy as a tool to visualize the molecule and to compare it to previous spike proteins from SARS-CoV-1, and at that time also the first structure of SARS-CoV-2 spike protein. So there are two steps involved. The first is to flash freeze these molecules in the ice and then visualize them by electron microscopy at very high magnification that we can see individual molecules. And the second step is we collect many thousands of movies of these molecules and use the different views we have in order to, in the computer, reconstruct a 3D structure at high resolution. High-performance computing is essential for modern cryo-electron microscopy because we record terabytes of movies of the molecules, and we deal with hundreds of thousands to millions of individual molecules which we visualize from different sites. Oracle provided us with cloud computing space and time, and rather than a local high-performance computer, we could use the cloud in order to determine the structure of the spike protein. So the key finding, and it is really a discovery for us because we didn't expect it at all, is the fact that in the spike protein structure which we solved, there is a pocket which is filled with a fatty acid, and it's not just any fatty acid, it's an essential fatty acid, linoleic acid.
Linoleic acid is an essential fatty acid. This means that it is something which the body cannot produce on its own, but which we have to take up by the diet. We discovered that this binding of linoleic acid stabilizes a form of this protein which is less infectious. This is a key finding, because if we can find now something which is even better binding than linoleic acid, and will lock down this protein in a completely non-infectious form, then we have defeated this virus before it could even infect us. Recent studies have shown that in patients who suffer severely from COVID-19, linoleic acid is depleted in the blood. So there are many hints and many indications that linoleic acid is a very important factor in COVID-19 pathogenesis, but until now it was not clear how these ends fit together. We have established with our discovery for the first time that there is a direct link between this important molecule and the virus itself and the disease. And this is now something which we need to look into, and we need to figure out how this can be used to improve treatments of COVID-19, but also to develop antiviral small molecules to defeat the disease. Everybody is talking about vaccines to defeat the COVID-19 crisis, and we all are hopeful that vaccines will be the solution. But there is a word of caution. HIV is a different, devastating disease. 30 years of research have not resulted in a vaccine against HIV. What has worked were small molecule antivirals. Therefore, we need many approaches to address the current crisis. We need to try everything to get the upper hand. COVID-19 is a global crisis, so we need a global solution. Antivirals could be this solution. So one immediate step could be to supplement linoleic acid in the treatment regimen of severely ill patients. In the longer term, we expect to develop linoleic acid mimetics which will shut down the virus entirely and be drugs themselves. What we now need to do is to assemble the team of experts who have the necessary skills and the funding to fuel this research and to make an antiviral development program happen. The University of Bristol and Bristol as a whole is fantastic to carry out this kind of research. We have coronavirus experts. We have the cryo-electron microscopy facility. We have a collaboration with Oracle. We have access to computing, and we have the entire ecosystem around the university, which make this the perfect place to do this kind of